Hello. Please hit like button and subscribe my channel. Also press bell icon for future video notifications. Thanks.
For a little more than a decade, scientists have been filtering water samples from aquariums, rivers, lakes, and even the ocean to obtain DNA that was shed by fish and other aquatic life. The goal? To use this environmental DNA, EDNA, to monitor aquatic species. Now, a trio of papers, 2 on animals, and 1 on plants, suggest it's also possible to detect and identify terrestrial organisms using EDNA floating in the air. Although the research, along with the entire field of EDNA, is in early stages, experts tell the scientist that the technology could make it more logistically and financially feasible to find and monitor rare, endangered, invasive, or shy species. Such studies will likely complement rather than replace existing monitoring methods such as camera traps, say scientists working with EDNA, but the ability to fill in the blind spots left by current methods could be immensely beneficial to ecologists. Genetic analyses including EDNA are a way of democratizing and enhancing our ability to know what's going on in the natural world, and also what we're doing to it. Mark Stokel, an environmental genomicist at the Rockefeller University who uses EDNA to monitor fish populations and was not involved. 2 of the 3 studies, both published today, January 6, in Current Biology, demonstrated the successful collection and analysis of airborne EDNA shed by animals. Those experiments, one conducted at and around the Hamerton Zoo in the UK and the other at the Copenhagen Zoo in Denmark, relied on the assumption that animals in pens, enclosures, and indoor exhibits would give off strong, consistent signals. The authors of both papers were able to detect and identify the DNA of dozens of different animal species. And in both cases, their results blew their authors' expectations out of the water, especially for proof-of-concept research. Based on their findings, the researchers conclude that animal DNA can travel much farther through the air than they expected. Both teams were able to detect zoo animals as well as those living outside the zoo, even from hundreds of meters away. We were seriously worried it wouldn't work, Claire tells the scientist. Lingard echoes that sentiment. When I was planning this, I thought of the worst-case scenario. Altogether, Claire's team was able to identify DNA from 25 different mammal and bird species that live in or near the zoo, as well as DNA from the food being fed to those animals. Sometimes a sensor located outside of a building would pick up identifiable quantities of DNA from a species housed inside, or from an enclosure located all the way across the zoo. There's something in the air. The studies follow up on earlier work in which Claire's team detected airborne EDNA from a colony of naked mole rats maintained in a laboratory setting, an environment with far fewer variables than the zoo. The perfect thing about zoos is you have all these non-native species you cannot mix up with anything else, Claire tells the scientist. And you also know precisely where they are. That became really important for both of us because we were picking up the animals we were near, the sensors, but a lot of other animals as well. See, environmental DNA can be pulled from the air. The zoo research is still considered proof of concept for terrestrial EDNA monitoring, though tacky. In this case, the 2 teams took a variety of approaches to collection, which the study authors say should be informative as airborne EDNA monitoring makes its way into applied ecological research. By contrast, Claire's team only used 1 kind of sensor and ran collections for, at most, half-hour bursts. They had tons of DNA, she adds, but because the other team ran fewer collections for longer periods of time, they didn't have as much detail on where it came from. For now, the process is far from perfect. Some animals living in the zoos, such as a tiger that Claire's team attempted to detect, were missing from the EDNA samples altogether. For now, any attempt to explain why some animals were more readily detected than others, Claire says, would be pure speculation. Two of the air collection devices used by the research team in Copenhagen, Christian Bendix, the tiger question, also confused Stokel, who didn't work on either of the new papers. He tells the scientist he would have liked to see more discussion of possible reasons that some animals went undetected, but is overall very complimentary of both zoo studies. When you're starting out, the positive results are the most important ones, he says. The negative ones are less important, and the positive results in these papers are great. Passively detecting plants. Meanwhile, research on airborne plant Edna is a few steps ahead, giving animal researchers hints as to what they might attempt next. Last month, Texas Tech University doctoral candidate Mark Johnson, his advisor, ecologist Matthew Barnes, and their colleagues reported in BMC Ecology and Evolution the results of a study in which they sequenced Edna from dust traps, which passively collect pollen as well as any other airborne mole. The team found several species of grass, fungi, and even an invasive species called tree of heaven, Ailanthus altissima, that had all been overlooked by more conventional surveys. Johnson and Barnes have conducted similar experiments before, but this paper looked at a year's worth of collection data, offering new insight into how seasonal changes, weather, and other factors impact the species detected by Edna, offering new insight into the ecosystem's dynamics. Other researchers are also trying to do the same with insect Edna. Preprint research presented at last month's Ecology Across Borders conference reportedly identified 85 insect species, and some vertebrates, from airborne Edna. We've shown that it works, now we need to try to understand some of the nuances of it, Johnson tells the scientist. How does wind, how does weather, how does height affect our collection? Leaving the lab as the field forges ahead, airborne Edna scientists do have one major source of guidance: the field of aquatic Edna research, in which researchers have several years' worth of a head start. Scientists working with aquatic Edna have already thoroughly demonstrated that the technology works and are now making strides toward using it as a standard ecological tool. In some cases, this work is already underway. Johnson is now working on follow-up research in natural environments that takes a closer look at specific variables such as distance, weather, and altitude, and a paper in which he uses his passive dust traps to collect animal Edna is making its way through. Unfortunately, that kind of research brings new challenges. We can't plug a water vacuum in the rainforest in Madagascar, Bowman tells the scientist. And also we can't make too much noise, which would disturb the wildlife. That's why her team tested a few different types of sensors, and why Johnson's passive collection research will likely prove valuable. We wanted something that would be transferable to a natural environment, Bowman adds. University of Guelph biologist Robert Hanner, who didn't work on any of the studies but helped shape the field of Edna research, says that the aquatic Edna research community still has plenty of challenges of its own. Although it has progressed further than the airborne Edna field, scientists don't. For example, ecologists are often interested in measuring the abundance of a given species in an area, and so far, aquatic Edna surveys only detect their presence. Much like Claire's issue with the zoo's tiger, Hanner recalls a researcher working in his lab who failed to gather Edna from a crustacean from water in its immediate vicinity. The challenge, he explains, is that the field doesn't yet know why that would happen. The conventional explanation would be that the PCR amplification somehow went wrong. For all he knows, certain sediments in the water might bind to Edna or the particles ferrying it, preventing collection of that DNA from happening in the first place. Yet these details often go unreported in the literature, which has primarily been saturated with proof-of-concept studies focused on showing that Edna analysis works at all. It's surprising how much we don't know about the natural world, even for familiar animals, Stokel tells the scientist. These new technologies are going to help us understand that better, and hopefully be better stewards of the environment. That's ultimately the goal, and in that way, I'm optimistic. Quote, Please support my channel to grow by pressing subscribe button and the bell icon. We will notify you technological news. Thank you.